Welcome back from the break. We'll now hear from Dr. Shivangi, who will provide us an overview of ion torrent sequencing, discussing the key principles of this technology and its applications in genomic research.
Thank you. Good morning, everybody. So it's like I've been a student of this lab for 2 years, and it's like coming home back, and I'm really fortunate to be a part of this meeting. So the topic allotted to me is ion torrent sequencing principles and overview. So we'll first revise the principle. It has already been discussed yesterday, but just to recapitulate and go a bit deeper. So basically, a chemically encoded information in the form of ACTG is converted to a digital information on a semiconductor chip. So this is kind of, we are again going back to the second generation sequencing from the fascinating futuristic fourth generation sequencing talk that we just had in the last lecture. So because most of us would be dealing with or would be given this platform to work on, so it's important to know about this. So when we see the principle, we all know that when a nucleotide is incorporated into the DNA strand or the DNA polymer, there occurs release of a hydrogen ion, and because of that there is a pH change which is recorded. And when there is no nucleotide which is incorporated, there will be no pH change, no signal will be recorded. And if there are two same nucleotides that are present, so the pH change will be double and will have a double voltage that will be recorded. So where is all this going on? So if you see this is a well, a micro well, in which there is a pink color sphere or a bead that is present, on which the template is already formed. So when we look at the overview, we'll come to know where this bead came from, where this template came from. But basically the unknown sequence that we are trying to understand or decode is present on this template. So against which the synthesis will occur. So as you'll recall, this is also a sequencing by synthesis, or the SBS method, in which the nucleotide will be added. So from where will they be added? So on this well, the flow of DNTPs will take place. So this is kind of known. So suppose here is a T and A is flown. So there will be incorporation of that A, and the H plus ion will be released, and the pH change will occur, and there are sensors here which are the semiconductor chip sequencing. So where is this well present? This well is present on something called as a chip. So this is occurring like Illumina we discussed, it occurs on a flow cell. Here there is a chip that is present which has these numerous micro wells, and in these micro wells these beads with template are present. So all these wells will have these beads, most of which are clonal, and so millions of beads with millions of wells. So that is why it is called as massively parallel sequencing. So when we zoom into the bead, so bead will have these templates which are mostly clonals. Clonal means, suppose here there is a sequence called ACTG, suppose. So all of them are supposed to have that same sequence. So this is a clonal bead. So when a T gets added here, so there are numerous T's that will get added, and accordingly, so this is being done so that the pH that is changing is detectable by the sensor. So that is why this clonal template generation is necessary. So the sequencing cycles or flows where known DNTPs are sequentially flown across the chip occurs, and the pH is detected by sensors. So these sensors are present in each of the micro wells present on the chip. So it's called something as embedded ion sensitive field effect transistor. And between the washes there are between the flows there are washes, so that there is no crossover. Like for example, we have a flow in which A is flown. So wherever there is a T, the A will get incorporated, the pH will change. Then there will be a plain wash. Then there will be a cycle of, suppose, T. So wherever there is A, the T will get incorporated and the pH will change. So everything is happening parallelly and the digital information is being recorded. So similarly for G, similarly for C. So this we have already discussed, like one nucleotide gets added, we know what nucleotide is there, so the pH change will be similar and the voltage change will be according. No nucleotide, no voltage change. Two hydrogen ion release, or when two subsequent nucleotides are there, double the voltage change. So again, so this we come back to what we were discussing. So well, bead with template, flows of DNTP, and the sensor in each well. So I hope this is clear. So the sensing technology is basically the same semiconductor technology that is used across digital cameras, gaming, LED lights, or other monitoring instruments, and even a rice cooker. So this is something which is, if somebody would have listened to Prime Minister Modi's talk 2 days back, so he flexed about it in New York that India is soon going to build this semiconductor units and all. So just a history of ion Torrent semiconductor sequencing, how these instruments came up. So first instrument was the ion PGM in 2010, followed by that in 2011 they got the AmpliSeq technology, which is the Amplicon based library preparation technology. Then in 2014 they introduced this ion chef instrument, which is the first automated template preparation instrument, which reduces the hands-on time. And then there were some assays which came up, which got CE, IVD approved, and FDA approval of drugs got when these assays were used. So this is how they gradually built up, and subsequently there were more machines. In 2018 there was Ion Gene Studio S5 system, and then in 2019 there was a Genexus system. So a lot of automation and automated library preparation and all came up. So in our institute we have been given Ion Gene Studio S5 system. So I'll be discussing the workflow based on this system. So everything starts with what and why. So the samples that you'll be looking on. So suppose, for example, we are starting with a myeloid panel. So AMLs, like we saw that there are so many fusions, and even the recent WHO has kind of a list of defining abnormalities which are very extensive. So it's always wise to have a DNA plus RNA sequencing, whatever best we can do at this stage. So according to that, we'll have the panels which we know, or the samples which we are going to proceed with. We'll then go away with we'll then go ahead with library preparation. It can be manual or automated. Automated one is very expensive. We are currently doing the manual one only. Then for preparation of template, we'll have to use this chef system of theirs, because that is like manual one is very like tedious, and almost nobody follows that. So after that, sequencing, what happens actually in the sequencing that we saw, and then there are they have their own softwares on which the data is loaded and the reporting and analysis takes place. So ready to use panels we can start with, like whoever is starting, but we can also design our own panel. So they have this tool of ampliseq.com/designer where you can design your own templates and have the panels. So just to look at how we proceed. So this is from the myeloid panel of theirs, which has both DNA and RNA sequencing. So for every sample you'll need to have DNA and RNA both extracted, and then of course RNA will need to reverse transcribe to a cDNA. And then, as this is an Amplicon based library preparation step, we'll first have PCRs to amplify these libraries, followed by the partial digestion that was discussed yesterday, the proprietary FUPA reagent, and then there are barcode and adapter ligation steps, and finally the equalization or combining the libraries. So the input of DNA that is used in Amplicon is less, so it's one of its advantages. Even for example in solid organ or the formalin fixed tissues, the input could be less, and this is the advantage of Amplicon based sequencing, that 10 nanogram of DNA or RNA is sufficient. So in this panel also, like we have 10 nanograms. So initially after DNA extraction we do a nanodrop to rule out protein contamination, and then quantitate in on qubit. So you know the calculations. So for example, you have a 40 nanogram per microliter suppose on a qubit, and you'll require 10 nanograms. So you will calculate accordingly, and then also 0.5 ml only will be required that ways. So after all that things, the target amplification PCR steps and other steps that we have already discussed, and finally the libraries that are prepared are 100 picomole, so it is close to 0.1 nanomole. So this is different from Illumina, so it is kind of little lower. And finally we'll dilute the libraries to 25 to 60 picomole before the templating occurs. So just a revision on amplicon based sequencing. So this is whole DNA that is present in the sample, and in case of targeted sequencing we are only interested in some areas. So the panel is designed in such way that using the primers against the regions of interest, and so this is region of interest and not the scary ROI that most of us can be like concerned with. So we'll do a PCR amplification step here to amplify the regions of interest. Then there are partial digestion of amplicons using the FUPA enzyme to remove the primers, and then there is adapter and barcode ligation steps. So this is the adapter and this is the barcode, which is patient specific. So suppose this is sample 1, sample 2, so barcodes will remain common, but the adapters will remain common, but the barcodes will change. And then we'll pool the libraries together. So there's also library amplification steps, and finally the libraries will be pooled together to go ahead for template generation. So template generation that we discussed, it occurs it happens in an instrument called as Ion Chef. So this is from our lab. This is Gene Studio TM S5 Plus system, and this is the Chef. So this is what happens in a chef. There is a kind of, we need to do something called as emulsion PCR that was discussed yesterday. So those beads that we discussed, they need to be formed. The template, the whatever libraries we have prepared, the fragments, they need to be clonally amplified on each of those beads. So this whole process, like it takes 14 to 15 hours. So suppose we put a chef at 5 p.m. Next day 10:30, 11, the reaction is complete. And so if someone wants to go back and read on emulsion PCR, so result of this template generation is that the chip that we have selected gets loaded with the ionosphere particles, the beads, with millions of these it should be templates, one in each well of the chip. So next question comes that we discussed about chips, like you saw. So which chip to select? So it will all depend on the output that you are targeting, the number of samples you're having. So now for us, example, we are a new lab, we might expect only 3 to 4 samples per week. So according to that, we need to know the throughput. So for example, myeloid panel, the RNA only 0.2 million reads are required, whereas the DNA panel we require 1 to 2 million reads. So we started with a 520 chip, which has the 3 to 6 million reads can be targeted, and then 4 samples we started. So accordingly, you need to know the chemistry, what is the whether it's a 200 base pair or a 400 base pair, and accordingly you can select the chips, plan on your run, how many samples are there, what is the chemistry, and what is the reads that you need to give according to the panel. So just so you get a whole range of scalability. So maybe when sample load increases, we can move on to the higher chips like 540 and 550 even. So what happens inside the sequencer that we have already discussed there, that the pH change gets recorded. So what happens after the run is complete? So after the run is complete, the data, whatever the digitally encoded data, it goes on to the Torrent suite software, which is present inside the sequencer. So just like, what do we need to monitor? So just like in Illumina, we are concerned with the cluster density, whether it's proper or not. Here we need to look at the loading, the ISP loading. So this red color is like a good loading, 90%. So if it's lower or higher, like first time, I didn't add that picture, but first time, the first run that we put, it was like only 22% loading. So there was problem in the library preparation, the dilution steps. So that was a blue color there. So this is the thing that we need to look at. Plus there is other metrics also. So it's all available on the Thermo Fisher website, whatever data is there. So whatever the raw trace calls are there, they are first formed into a .dat files. So whatever the pH change or the digital information, and then in that also there are processes in which it will see that whether there are empty wells or not. Then there can be some polyclonal beads also which can be present, so all beads are not going to be clonal. So all that filtering it does, and then subsequently proceeds, and then dot wells files is something which is generated, and ultimately the BAM file is finally formed. So here FASTQ doesn't form directly, unlike in the Illumina platform. So this whole data which is in the Torrent suite software, it needs to be uploaded on the ION reporter software, and then there the data analysis, further data analysis and annotation processes takes place, and the report generation. So we are still in a process of exploring this software. So finally, the advantage of Thermo Fisher or the amplicon based sequencing is that a smaller input is sufficient. We get faster results, there's scalability, but the limitations of course are there, or the concerns are for homopolymer repeats and all are raised. But there are some algorithms that they claim that it's taken care of, and the error rate does come down. So this is a small journey I thought I'll share. Like we are starting with 2 assays. And since Jan, we are also doing regular PCRs. So initially, these were all outsourced from our state, and we are doing them in-house. PML report, we are almost providing the same day or the next morning. And these are some glimpses from our lab, and this is my team. And many thanks to Active Molecular Hematopathology. So this is the place that I learned and trying to implement my training out there. So thank you. So any questions or?
I think Shivangi, a common question that people have, because you have seen in our lab Illumina data and now you are seeing ion Torrent data as well. So how will you, because many people who are starting will have this doubt, right? Like whether they should go for Illumina or ion Torrent. So what will you think? What will you say about that?
Yeah, so I would prefer to be neutral, because every platform has its own advantage or disadvantage. And coming to library preparation steps, I think it's more or less the similar amount of effort that goes in. The number of steps would be lower in an amplicon based library preparation. Coming to data analysis, of course it's more like user friendly kind of, so everything is automated, so we don't have to put those manual efforts. But of course there are drawbacks also to that. If like we don't know what, so we'll have to like depend on them for the bioinformatics support as to if something is right or wrong, so to realign or kind of go through the bioinformatics again. Yeah, troubleshoot, exactly. So
any more questions?
okay, yeah, thanks.
Okay, so the next session is pretty special. We have a session on quality control basically, but we have an ensemble cast like what you expect in maybe a Quentin Tarantino movie or something like that. This is a cast of our lab's PhD students and many research fellows. So all of them will combine together to give you this talk. So please welcome. I welcome Anuradha, who will be the first one. Yeah.
Thank you, sir. So good afternoon, everyone. Me and my team members are going to give a group talk on quality control and NABL documentation for genomics. So let's get started. First of all, let's see what NABL is. So NABL stands for National Accreditation Board for Testing and Calibration Laboratories. It is an autonomous body under the Quality Council of India, which provides accreditation to medical laboratories in India. It is a third-party assessment which assesses the labs for quality assurance and competence and follows ISO 15189-2022 guidelines. It has an international recognition as it is a signatory of APAC and ILAC organizations via mutual recognition arrangements, which facilitates the global acceptance of test results between partner countries. So, accreditation means a formal recognition of technical competence, which ensures authentication of reports and gives confidence to customers and credibility to the lab.
This whole accreditation process is voluntary, means it is not a compulsion on the labs to go through the NABL accreditation procedure.
Anuradha, we have lots of time, so you can go slow also.
Yes,
Lot
okay.
of time.
Yes. So, NABL has a set of clauses which laboratories should follow. These are management clauses, these are technical clauses. So, starting with management clause, the medical labs should have a lab director who must have the qualifications, competence, authority, and resources to meet management and operational requirements. He or she should be responsible for implementing the management system, applying risk management, and ensuring continuous improvement. All the duties must be documented, of course. And the director can delegate duties to qualified personnel but retains the ultimate responsibility of handling the things. But delegations also must be documented. Moving on, next is the laboratory activities and structure clause, where the labs must facilitate communication with the users about examination choices, interpretation, diagnosis implication, and further actions via direct contact or email or etc. The laboratory personnel are encouraged to participate in clinical rounds and meetings. The management, technical, and support roles should be defined properly, and there should be a definite personnel for ensuring the quality management is properly taken care of to ensure effective operations. Moving on to personal requirements. So as the quote says, that teamwork makes the dream work. So personal requirements is a must and mandatory procedure to take care of for recruiting the right staff. It should match the range, volume, and complexity of tests in lab, and they should follow a compliance with national, regional, and local laws in that is mandatory thing. And the competence assessment should be documented, and training should be done properly to handle multiple responsibilities, as well as the regulatory compliances like biomedical waste disposal. All the clinical decision rules must be framed and verified by medically qualified personnel. Next clause is facilities and environmental conditions. There should be adequate space to ensure sufficient space for all lab function and avoid cross-contamination. Proper safety measures should be taken care of to ensure staff safety from different injuries. Exhaust systems should be properly installed if the labs are handling with chemicals like formalin, acids, and there should be an uninterrupted power supply for uninterrupted lab operations, and the equipments like centrifuges should be placed separately from the testing instruments to avoid interferences. Now, these are the major equipments that we use in an NGS laboratory. All of these equipments must be verified before use, and the verification includes installation qualification, operational qualification, and performance qualification, which includes minimum checks for accuracy, precision, linearity, and carryover tests. The IQ and OQ are performed by the manufacturer of the equipment, and that costs extra to the labs, but that is a mandatory to do thing for the labs. And the PQ is done by the labs itself, and that is done periodically, and all of the things are properly documented. This is how we put the label on different equipments, recording all the important information that has to be maintained properly on the instruments. So all the labs also should have biosafety cabinets that are important to protect workers, samples, and the environment, the working environment, from exposure to biohazards. Our lab uses biosafety cabinets class 2A and biosafety level 2 safety protocols to ensure the safety of workers, samples, and environment. And all the performance verification of biosafety cabinets should be done upon installation as well as annually thereafter. The equipments like water baths, incubators, refrigerators, which are temperature controlled, must be regularly verified with calibrated temperature recording devices, and there should be an arrangement for continuous temperature monitoring for critical equipments like refrigerators and freezers. And of course, maintenance of all the equipments is a must, and repeated checks on power supply as well as critical components like photometers and pipetters is mandatory. So now I'll hand over to the next speaker. Thank you.
Being a part of the diagnostic lab, calibration is foremost
Introduce yourself,
important.
please, Payal. Introduce yourself and then talk. Just
Okay.
tell us who you are.
Good afternoon, everyone. I'm Payel Gupta. I'm a PhD scholar under Dr. Nikhil Patkar. So I'll be taking over the next few slides. So being a part of the diagnostic lab, calibration is foremost important. It ensures accurate and reliable performance of the analytical equipment, and it adheres to the manufacturer specification and regulatory standards by NABL. The instruments that are present in our lab include automated and semi-automated like NextSeq, 1000, MiniSeq, MySeq, PCR machine and analyzers, and some non-analytical like centrifuges and pipettes. The key calibration activities include verification of all essential components like power supply and temperature controls. It should not just be the calibration or confirmatory statement. We should also keep a record of all the raw data generated during calibration activities. Calibration depends on the user's frequency and risk assessment. Generally, all instruments undergo annually calibration, but again it depends on the user's frequency. For all reagents and consumables, clear description of the material should be mentioned. We maintain a proper record of the date when the reagent was received, as mentioned, as seen here. Also, we also keep a record of when the reagent was opened. The storage requirements we make a point to ensure that it is maintained at a proper temperature, and if it is light sensitive, we also take that into consideration. Any specific handling instruction related to expiry is also taken into consideration. Being a part of the diagnostic lab to maintain uninterrupted diagnostic services, the institute has a policy to establish rate contracts for all the products, which ensures that we get them on time and at a lower price. We also receive samples from other institutes like Varanasi and Mulanpur. Hence, the institute at both ends have a policy to transport the samples, like use of proper temperature loggers, and they are transported upright without any leaks, including ice packs to maintain again proper temperature. This is especially important to prevent any RNA for the RNA based test. Thank you. Now over to the next speaker.
Okay. Good morning, everyone. Once again, I'm Neha Ghosh, and I'm a PhD student under Dr. Gaurav Chatterjee. Let's begin with pre-examination processes. So, starting with sample collection, the EDTA tubes are preferred for blood samples to avoid the use of heparin. And actually, we avoid the use of heparin because it can inhibit the PCR and it will negatively impact the NGS results. As for DNA extraction, blood can be stored at room temperature for up to 24 hours or at 2 to 8 degrees Celsius for 72 hours. For RNA extraction, it is crucial to perform the extraction within 6 hours to maintain the RNA quality for RNA sequencing. And if the extraction is delayed, it is recommended to stabilize the RNA immediately. As for records, in our lab, we aim to extract the RNA within 24 hours to maintain the maximum quality. For tissues and body fluids, snap freezing or immediate processing within 24 hours is necessary to preserve the nucleic acid integrity, which is critical for the NGS library preparation. Tissues can also be preserved as FFP, that is formalin fixed paraffin embedded blocks. During shipping, especially over the long distances, samples should be transported on dry ice. If processing is delayed, storing samples at minus 80 degrees Celsius is crucial to prevent the nucleic acid degradation, which could also compromise the NGS results. And when we are dealing with cell-free nucleic acids, which are essential for the liquid biopsy NGS assays, specialized tubes should be used, such as the Paxgene tubes, to prevent the contamination and degradation. Nucleic acid extraction from plasma samples should be performed within 4 hours of collection, which is sometimes not possible since we receive samples from we receive from Payel also hospital. So the plasma should be separated as soon as we receive the samples from the blood cells within 6 hours. And plasma can be stored up to 2 to 8 degrees Celsius for up to 5 days. However, for longer storage, minus 20 degree or minus 80 degrees is recommended to maintain the cell-free DNA quality. I'll move on to For nucleic acid storage for NGS, DNA is best stored at tris EDTA at pH 7.4 to stabilize it for library preparation. RNA should be stored in RNase-free tubes to avoid the repeated freeze and avoid repeated freeze-thaw cycles to preserve its quality for RNA sequencing. And nucleic acid concentration should also be quantified using fluorometry, that is a qubit count. For sample transportation, the collected sample should be transported to the laboratory under adequate transport policy, the hospital transport policy. Factors for transportation also plays a major role in molecular testing, such as the temperature during transport, the transportation time, also the placement of the samples in a transport container. And it is suggested that the specimen should be tightly sealed in plastic leak-proof containers and in this leak-proof plastic bags, as you can see in the image. This is how we receive samples when they are received in our lab. Next, I'll move on to examination processes. In NGS library preparation, ensuring the quality of nucleic acids is very critical. This is achieved by amplifying the housekeeping genes to verify integrity before proceeding. During fragmentation and amplification, validated methods must be used to ensure the consistent DNA-RNA fragmentation, which is essential for accurate NGS data. And electrophoresis should be performed to check the fragment size distribution also. For PCR amplification, careful primer designing is very critical. Performing a blast or a BLAT search helps to avoid the off-target amplification, which could intervene with the NGS data quality. In-house assays must be validated for NGS compatibility with a focus on specificity and reproducibility, and all necessary conditions must be documented to align with NGS workflows. And amplicon size and purity should be verified using gel electrophoresis. In cDNA synthesis and RT-PCR, appropriate primers such as oligo-DT or random hexamers or gene-specific primers should be used to convert RNA to cDNA. This process must be carefully controlled as it directly impacts the NGS data quality. Finally, in quantitative PCR, that is qPCR for NGS, generating a standard curve is essential for accurate quantification of targeting sequences. Thank you. I'll hand over to the next speaker.
So hello everyone, my name is Aarti and I'm working as a senior research fellow under Dr. Patkar. So I'll start with examination processes. So in this case, so this workflow involves sample preparation followed by nucleic acid extraction, which leads to library preparation and which is followed by sequencing. So after each crucial step, that is extraction, library construction and sequencing, the quality control has to be checked properly for the maintenance of the reliable data. Then we come to the PCR processes. So the validation, the proper validation and documentation of the post-PCR systems like gel electrophoresis and capillary electrophoresis should be done properly and it should be maintained. It also should be available for review. Also, appropriate controls to be used for each run. Laboratory shall demonstrate that the selected instrument has attained internally and the level of specificity is adequate for detecting the expected products. in case of PCR product storage, unlabeled PCR products can be stored for maximum 72 hours at 2 to 4 degrees Celsius. Fluorescently labeled PCR products or cycle sequencing reaction should be stored at minus 20 degree for maximum 48 hours. Also, care should be taken when using the intercalating dyes like ETBR for visualizing PCR products by using personal protective equipment. For example, in our lab, we are strictly following the biomedical West guidelines. And appropriate care and biomedical West guidelines should be followed for disposal of cytotoxic material. Also, alternatives to ETBR like CyberSafe, Gel Red, or Eva Green can be used. DNA sequencing analysis, validation of each technique to be done using known mutation as well as normal samples. Also, results of validation data should be well documented and it should be available for review. Laboratory must follow a GWS nomenclature for reporting genetic variations. During assay validation, testing is undertaken to determine the lower limit of detection. ACMG guidelines should be utilized for detection of germline variant and also interpretation of hereditary disease. AMP recommendation should be followed for the categorization and interpretation of somatic variations. In case of post-examination processes, the samples which are for infectious disease in molecular testing should be stored for 1 year at minus 80 degrees Celsius. Clinical sample for other molecular testing can be stored for 5 years at minus 80 degrees Celsius. In case of recommendation for NGS analytical wet bench processes, the identification of relevant sample needed for the test can be maintained properly. Also, documentation for acceptance and rejection criteria for different type of specimen can be recorded. Validation should take into account the issues relevant to all sample types and nucleic acid isolation methods. I'll now hand over to next person.
Good afternoon to one and all. My name is Karishma Anam and I'm working as a Junior Research Fellow with Dr. Nikhil Patkar. So moving on to the next slide. Library construction and enrichment. As we know, we are going to validate our library in first hand. So during the initial validation phase and verification phase, how we are preparing our library need to be documented properly. Which panel genes we are going to include in our library preparation need to be documented and validated properly, and technical SOB should be maintained throughout the library construction. Moving on, the use of barcode and indexing and its protocol for sample pooling should be maintained very carefully, because if we have two samples, that is if we are pulling more than two samples in a NGS run, as we know we can't sequence only single sample, we need to pull multiple samples for sequencing. So if in case we are having 96 plate well indices, and if we are using same indices for two different samples, and if it goes in single run, so if the qubit count of one sample is 9.38 and the second sample is 1.52, so during sequencing, the whole reads will be generated for sample 1 and sample 3 data will not be generated as they have both same indices they are having. So we need to carefully cross check before sequencing. If there are same indices for different samples, these samples need to be sequenced in two separate runs. If we sequence in the same run, one data will be generated and second data would not be generated, which will lead to failure in one data generation. Second is the library count and QC for how much the peak size is there. So if we want to sequence 300 base pair paired and library, our sequencing peak should come around 300 base pair. If there are some peaks before 300 base pair, they will take our reads counts, and the allotment for the specific library reads won't be there. So if you are having adapter dimers, double cleanup is necessary. So this quality control check is necessary before sequencing any run. Moving on, on target information like detailed description of analytical target region should be maintained properly, as if there are off target region that are sequenced greater than on target region, it will lead to lesser number of coverage, which will lead to lesser number of WAF region, and that will lead to missing of mutations that are very crucial and which has lesser WAF. If you want a great median coverage for our lab generally is 200x, greater than 150x. So if there are off target, on target should be greater than 60%. If there are more off target region, it will lead to less number of median coverage. In first example, there is coverage are more than 200x coverage. So this sequencing run is the pass run. If there are median coverage is less, then this is the flag run. We need to repeat this sequence run. Moving on, with the run, we need to have a control sample. If there are any contaminant in our reagent or environmental contaminant, that can be excluded by using a control run. So after our data is generated, we can simply nullify control sample or normalize our contaminants from our samples, and we can get exact count for our sample mutations. Tracking of the manufacturer versions of the sequencing reagents and disposable also needs to be maintained, like if the kit is expired, which may also lead to failure of run. So inventory of kit, when it came, when it arrived, what is the expiry date, and everything need to be documented properly. The acceptance and rejection criteria for sequencing data, which is generated after the sequencing run is completed, it should also be validated and documented properly. And it can be validated using metrics and quality control parameters, which are established during test optimization. This would be continued by next speaker.
Hi, good afternoon. My name is Elwin Paulose and I work as a JRF under Dr. Gaurav Chatterjee. So I'll be briefing on recommendation for NGS analytical dry bench processes. So the first point is sequence alignment for variant calling. So the main objective is to evaluate sequence alignment quality to ensure accurate variant calling. So basically, there are three key quality measures. The first is mapping quality scores, the second is mapping fraction, and the third is the base quality score. So mapping quality scores and map fractions cannot be used interchangeably, because map quality scores am I audible back? Yes. So mapping quality scores and mapping fraction cannot be used interchangeably, because mapping quality scores talks about a particular sequence aligning to your reference, as compared to the mapping fraction, which is the entire region or the entire genome aligning to your reference. So basically for mapping fraction, the NABL states that defining acceptable percentage alignment. So in our case, in our lab, what we do is we accept the alignment score of 97% and we use various alignment depending on the assays, such as bow tie or BWA. And the third important point is the base quality score. So for base quality score, I have put a screenshot of a QC report for base pair sequence quality. So basically on your x-axis is the quality score, which is the Fred score over here, which is denoted by Q2 to Q34. So basically the green region symbolizes good quality basis. The yellow region symbolizes approximately medium quality basis, and the red one is the poor quality basis. So over here, what in our lab we keep a bench cutoff of Q30. So Q30 signifies one error in 1000 base pairs, which will be elaborated in the further sessions. So what we do is, over here is the base pair positions over here, 1 to 151. So what we do is we take a cut over here, which is around 120. So these are basically your good base quality scores. And ahead over here from 130 to 151 is basically your adapter, which will be trimmed out further by using adapter trimming software such as Trimomatic or Cut Adapt or Fast Speed, depending on your assays. So over here on the left side, what you can see is this is basically a screenshot of FastP which I have put over here. Basically in our lab we use Trimomatic software. So over here on the left side, what you can see is the FastP reports over here, which shows this is the adapter content. So when you can see from here from 75 to position 139, you can see adapter in this sequence. So you need to remove it. So over here you will get adapter content cross. That means there is adapter contamination in your reads. Any questions in between will be entertained. Please be free to ask. And on the right side over here, you can see is the adapter content is like tick, means no adapter contamination can be seen in the sequence. So the next one is variant calling and validation. So the first point is bioinformatic pipeline, detecting clinical relevant variants such as SNVs, indels, and CNVs. Variant types and size limits, you need to define the largest and smallest detectable indel and CNV sizes. So for validation process, you need to use the HGVS nomenclature for reporting. Focus on 3' position of reference sequencing for indel representation. Validate the pipeline detection reliability for each variant type and identify limitations. So the next point is analysis of VAF. You need to define your VAF ranges based on the heredity of disease. So for hereditary disease, identify VAF ranges for heterozygous and homozygous states. In heterozygous condition, basically you have one allele affected, which is equivalent to 50%. So based on the technique which you're using, the VAF ranges might be to 30 to 70%. You need to standardize on that. And basically on homozygous state is both the alleles which are affected, the VAF should average out around 100%. So for second point is for acquired cancer and mitochondrial disorders, you need to define low VAF detection limits for these conditions. For key consideration, VAF for cancer and mitochondrial disorders can range from 1% to 100%. In our case, for limit of detection for baseline or untreated patient, we have a cutoff of 3%. And on MRD time point, it is for 0.03%. So you need to establish guidelines to manage somatic mosaicism and distinguish low VAF from instrument errors. For internal guidelines, define condition for additional verification to ensure accuracy in low frequent variant detection. So the last and the most important point is for data storage and retention. For file retention guidelines, for VCF files must be retained for at least 5 years. In case of minors, retention can be extended for 5 to 10 years. For raw data retention, FASTQ, SAM, BAM should be stored for at least 5 years. Consideration for longer retention should be based on the annual data turnover, and the laboratory's ability to handle big data retention periods may be extended. Further talk will be continued by my colleague. Thank you.
Thank you, Elwin. Myself, Priyanka Ugule, and I'm working as a JRF under Dr. Nikhil Patkar. So, coming to the next point, recommendation for interpretation and reporting of NGS results. So, the things which are learning from yesterday and we will be learning for the next 5 days, that is for what? Giving out proper results, right? To enhance the accuracy, clarity, and clinical utility of clinical NGS data and reports, it is very necessary to adhere to the established guidelines and best practices. So, what should an NGS report consist of? So, before giving out reports and results to the patients, we should learn that the NGS report should consist of the following points, such as patient's demographics, the number of genes covered in the panel, details of the tests, details of the variants identified, details of the techniques which are done, interpretation including variants classification. Also, it contains the details of the laboratory and the clinicians responsible for the report. So, coming up to the next important point, variant interpretation and reporting. So, variant detection should be done to identify many genetic variants. There are several computational tools that predict the functional consequences of variants. After determining the likely impact of a variant, it must be correlated with the patient's data. So, human sequence variants must be reported using HGVS nomenclature as well as a standard version reference identifier to the transcript or protein, that is RefSeq, accession number, ensemble transcript, etc. that enables unambiguous mapping of the variant. The laboratories may follow updated ACMG guidelines recommendations as applicable from time to time for reporting a specific number of genes, or design their own reporting policy with availability of the informed consent of the patient. So coming up, we can see a report which our lab generates. The main important things we can see are patient demographics at the very top. Next thing is the number of genes covered in the panel, which is mentioned. Next, in the interpretation, we can see the name of the genes, genomic variants, protein variants, and the other details. Next, followed by the clinical interpretation, which is done by the medical practitioners. Next, in the later half, we mention the workflow, that is the techniques we use in the laboratory to give out the result, and the proper table of the gene panel used. Reporting of incidental or secondary findings. So, it refers to the genetic variants that are discovered unintentionally while sequencing for another primary reason. These findings may or may not be related to the patient's original diagnostic question, but may have a significant implications. So, what are the key considerations to be noted? So, A. Incidental findings. So, incidental findings include variants that are found unintentionally during primary NGS analysis. B. Secondary findings. So, the secondary findings include analysis of the predefined set of genes known to be associated with the significant health risks regardless to the primary reason of sequencing. So, for effective interpretation and reporting of the next generation sequencing data outcomes, improve the accuracy, consistency, and clinical relevance of the NGS variant interpretation and reporting, ultimately leading to the better patient care and outcomes. So, next will be continued by my colleague.
At times, I do come across cases where I have a discrepancy between the AMP classification and the ACMG.
Yeah.
Some variant is called out as likely pathogenic and is a tier 3. So how do you come to a consensus for such variants?
Yeah, so firstly that we look at ACMG only if we are doubting a germline inheritance. So if it's close to 50%, 40%, 50%, then only we start looking at ACMG. So what AM does is like somatic mutations, clinical importance for that gene. So it could be a gene, could be pathogenic, but it may be not somatically important at that particular disease context. So we have had a lot of discussion about that. So for example, let's say TET2 mutation in AML. So we know that it should go maybe tier 2 because it doesn't have a clear-cut diagnostic or prognostic significance, but maybe it is tier 2. But there may be specific subsets of AML where it is more important. So if you're looking at that subset, should you upgrade it to tier 1? So this kind of question you always have. But what I will just say is that AM tires and ACMGs are different. So one gene could be pathogenic according to ACMG, but still its clinical importance might be.
So how will we ultimately take a side? What should I ultimately report it
So
as?
if we think
if they are frame shift indels, no, most of the times the ACMG classification will be like likely pathogenic.
Correct, yes.
And AMP will give it as tier 3. Most of the times I've seen this happening, and I find it very
So if I know that that gene is implicated in that disease, I will go with tier 2 then only. Because for indels it is again slightly different. For indels there might be.
For example, Abhi, recently I have a case who is a suspected case of polycythemia vera. All the driver mutations are negative, but he has a in-frame insertion 9 base pair in ZRSR2 gene, and then that has been called as a VUS.
By
tier
ACMG.
3, that has been called as a likely pathogenic by ACMG and AMP me tier 3. So what should I ultimately? It's a VIP patient, so you give me a clear-cut answer.
So for like ZRSR,
I'll send you that Excel file.
see the ZRSR2 mutation itself in the context of polycythemia vera, we do not know the importance of it from that extent. So I will go with tier 3 only because what is the even if it is pathogenic,
I wanted to hear that.
whether it is somatically important in MPN. So these are two different questions. One is looking at if it is pathogenic or not. The other is looking at if in a particular disease context it is important or not. So that way we can answer that, I guess.
To add on,
Yes.
as a question, like for example, like sometimes we find a variant that is not in the cosmic database.
Correct. All the time actually.
So during that time, but it is an important gene, and then we know like that the variant that we have got is associated with the disease mechanism as well.
Yes.
So during that time, how to take it?
So we will discuss some of the examples in Saturday session, like how you are, I think, mostly talking about from ACMG perspective here, right? Like it's close to 40-50%. So then how to tackle that, we will discuss a bit. But basically you have to, I think the good idea is to go individually with the codes rather than just looking at, what we do is we look at aggregator softwares, right? And then whatever they are saying, we kind of go with that. it's a good idea to use that, but at least what codes they are providing, we should probably check them individually and then take a call, like in the as a whole what it is. So then if you think this is VOS, you should go with VOS. If you think according to ACMG it's pathogenic, you should go with pathogenic, I think. And then if that gene is important in that disease, then it automatically becomes either tier 1 or tier 2. Like the moment you have decided that it is pathogenic, it could be tier 1 and tier 2. But again, for the germline variants, like what you're suspecting as germline, you need not give AM tires also. You can just stick to SCMG. This looks like pathogenic, and you can stop there because this is anyway a germline variant, right?
Then like mostly I work with a solid tumor.
Yes.
Sometimes like I get the solid tumor with the tumor percentage of 80%, and I get a VAF of 50%.
Right.
So
pretty high. Yes.
So that time like it comes in the mind that whether it is like truly a germline.
Yes.
Or it is a somatic.
So those cases, it's good idea to do SCMG. But then again, whether that gene has been reported in that particular disease to have germline inheritance, those things also are important, because sometimes it could be just a clone has dominated everything and it's probably somatic only. But literature is also important. So it's not an exact science. Definitely it's not an exact science. We also, in many of the examples that we will show you on Saturday morning, so we have had problems, we have had discussions at length, like maybe something that I would say Shweta will not agree or Dr. Nikhil will not agree. That will happen, but that is how it is, unfortunately. Yeah. Why not go for?
Yeah. Recommendation.
Yeah. Again, our country still we can. Of course, we do always recommend that. Like you do a buccal swab, or you do a peripheral blood in remission, or you do parents. So we always recommend that. But at that point of time, you have to give something in the report, no. And sometimes it's also not possible. Like it's not always feasible that the parents will come and give samples. That is feasible in our country. In the West, they will not allow only that parents to give you sample. You need to go with either buccal swab or peripheral blood. Of course, there. You need to establish the germline inheritance only by testing, not by assuming. Yeah.
And also, I think there is recent update that we have to nowadays stop following this TS system and we have to go for point system.
Yeah, definitely.
It is going to come in next 6 months.
Yes. Not yet there, but it is getting in. It is getting ready. Yeah.
Good afternoon all. I am Komal Kumbhalwar and I am the PhD scholar under Dr. Naveen Khatri, Dr. Nikhil Patkar and Dr. Sachin Punatar. So in next few slides I am going to share some glimpse on the recommendation for verification or and validation of NGS test. So the performance and the documentation of NGS test validation is central to assuring the quality of NGS test. The purpose is to address the accuracy, precision, sensitivity, specificity and the limit of detection of the assay. So in US, it is the regulatory requirement under the Clinical Laboratory Improvement Amendments Act 1988. In other countries, it is based on the standard set by ISO 15189. So as per the NABL guideline, so minimum of 20 specimens, mix of positive, negative and the reference standard should be included in the validation. For designing the validation protocol for NGS, so it is important to consider all steps in the validation process, including nucleic acid extraction, enrichment, library preparation and the data analysis. And it is important to establish threshold for the alignment process to flag reads that are not considered high enough quality. Establishment of accuracy by considering several NGS parameters, including depth of coverage, number of reads and the Q-score. It is important to establish the robustness and the reproducibility to ensure the consistency in the performance independent of operator and the reagent lots. So the assay validation should include the outline protocol in which the performance specification should be established for this performance metric, which includes the accuracy, precision, analytical sensitivity, specificity and reportable range and reference range. And in CLIAS, the definitions has been derived for specifically for NGS for these performance metrics. So accuracy is the degree of agreement between the nucleic acid sequences derived from the assay and the reference sequences. The precision includes the degree to which repeated sequence analysis gives the same result, that is repeatability within run precision and reproducibility, that is between run precision. It is important to establish the analytical sensitivity, that is the likelihood that the assay will detect the targeted sequence and variants if present. Limit of detection and the true positive rate is useful measurement for the analytical sensitivity. Analytical specificity is the probability that the assay will not detect the sequence or the variant when none are present. The false positive rate is useful measurement, and also it is important to establish the reportable range and the reference range during the assay validation. So next I will be brief on the external quality assessment. So it is also referred as proficiency testing, and is important to monitor the laboratory performance of examination methods by comparison with results of other laboratories. Laboratory should participate in EQA programs prior to gaining accreditation in case of change in test methodology, equipment changes, and the exchange of scope. So with the performance, the documentation of corrective action taken based on the EQA evaluation report is also crucial. In this slide, briefly I will explain the process where the EQA samples are sent regularly to the participant laboratories, where they analyze the sample, they generate the results, and it is shared back to the EQA organization or provider, where they evaluate the lab reports and perform the lab specific performance report, which is shared back to laboratory. So depends on this evaluation report, if there is any non-conformance, if applicable, corrective action should be taken, and it is important to document the same. So there are some important interlaboratory comparison EQA programs, which includes the UK National External Quality Assessment Service, College of American Pathologists, that is CAP, European Society of Human Genomics Eurogenus program, and the ERIC guidelines. For some analytes, there are UK services are not available or not feasible. In that case, the laboratory should adopt the alternative approaches, which include the interlaboratory exchange or split sample analysis with other laboratories, exchange of blind samples with other laboratories, analysis of DNA from cell line with the previously determined genotypes, and can also use the analysis of a synthetic DNA of known genotypes. Thank you.
Yes, ma'am.
Hello. Gaurav, as far as the proficiency testing is concerned, we are doing it with your lab. So for DNA-seq, RNA-seq and RQ, it is very sorted. But as far as the KDN goes, we do it by Sanger sequencing and you do it by NGS.
Yes.
So that will not work out, no? Because we are not working on the same modality. So the another thing that we did was we tested the same sample with Sanger and run it on a DNA-seq, and we got the same kind of mutation. So will that suffice, because we are going for NABL in December? So should we send it to you, or just as a safety sake, or will this suffice?
To me it will suffice, but I think
you tell me, then I'll send it to you, na? Because the only worry is
Neha, I just want to interrupt.
Yes, please. What do you think?
Yes, because right now in the system also they have stopped the UK ANIQUA service and the CAP service.
That's also true.
So for that cases, you can do the split testing as it is applicable for NABL. So if you have it, you can do your own testing if it is not available anywhere. And in Sanger sequencing, the thing is that if you are sending samples to us and it is not running in our laboratory, it is little difficult to whatever the results we are giving and you are doing it.
Obviously, if you are doing it on NGS platform, you will get more number of mutations.
Yes. But at least whether if you are detecting those or not, that is we could do it for safety. Say you can send it. We can do it.
Okay, so one I'll just send for safety.
Yes, you can do it. But the thing is that previously what we were giving in Sanger sequencing, we were giving the frame shifts and all.
Correct, yes.
Right now we are giving only the mutations.
Let's have a look at it.
That is why we cannot be on the same page if we are giving this.
But we can have a look at it.
No problem that it should not fail.
No, in that cases you can do split testing, means you can give the test to two of the persons which you are having it.
One is done by Nilesh, the other is done by Rohit. So it is
so
the
then
proper
whatever
kind of
the results is there, you can
on different platforms also.
Yes. So that
to me
is
it will
okay.
suffice.
That is acceptable ones.
And now do you report the splice variants anymore?
No. We have
so
stopped
we should
it
also stop that.
Yes, but it seems like it's a population level alternate splicing in our data set. Yes, Karthik.
Yeah. If we are getting kits which are supplied by companies and is wet lab validated by them, and we're following the same protocols which was given by them without any other tweaks, so then in that case also we have to do the LOD LOQ assays.
And don't
or directly go with the company.
If you are completely following their instruction and only in the context that it has been approved for, I don't think we need to do that. But if you are using it for some other purposes, then probably it's better. Anyway, it's a good idea to do it in your lab. But for documentation purpose, for example, clonoSEQ is approved for MRD testing in myeloma or BLL. So in that context, if you're using that FDA approved kit, I don't think we require to do a validated LOD-LOQ again.
No, it is not.
We don't.
And then for your MRDs,
Yes,
what is your VAF you're going reporting is 0.02.
Yeah, so it depends on test to test. So for I think our FLT3 it is slightly more sensitive, so it's 0.003 I believe, and for others it is 0.03. As
So for those,
the fellows will know better.
The LOD-LOQs you have done for those cases.
Yes.
Right.
Yes. Any new test you are, for example, even if we have done, so for example, from MIPS, if you are shifting to a amplicon-based assay, then we again have to, ideally you again have to do LOD-LOQ. So any change you are doing, you should again establish
And that
LOD-LOQ.
should be properly documented.
Like in the flow cytometry context, just to look for the machine's capability to pick up this.
Yes.
Because everything depends on the sample heterogeneity.
Yeah, here it is not like that.
So
Now here the processing thing, if it changes, it might change. Your LOD-LOQ will change. So then you should do it again if you have changed your
My
test.
thing is that like LOD-LOQ assays, you do it in a controlled environment in a known
Yes.
sample.
Yes.
So you have to make sure that your machine is robust enough to pick up to that deep level.
Correct.
So in a given sample which is coming day and day, the hemodilution, those things are not really counted.
That is enough. That is enough.
That is
You can
enough.
do it. Yes.
Okay, so we have a early lunch, I believe.
Okay, so I kindly request everyone to make their way to the cafeteria for lunch. And also after lunch at 2 p.m. we'll resume with our wet lab sessions.
Should we 2 or should we
start
prepone it
by
a bit because we are early, no? I think it should be 1:30.
Yes, 1:30.
Yeah. So we assemble in the lab at 1:30. Yeah.
So that we can start our work properly.
And today I think we'll follow what exactly it was yesterday, right? So whoever was in wet lab is in wet lab. Whoever was in bioinformatics then in bioinformatics.
The group is already decided.
Yes.
The group A and B will go for the wet lab.
We'll switch
and
tomorrow,
C
I
and
believe.
D. Tomorrow we will switch
Yes.
it. And it is already mentioned on your pamphlet also.
Yes. So 1:30 in the lab. Please enjoy your lunch.
Yes.
Thank you.
Also a humble request to everyone that please